Hi, my name is Vanessa. Today I'm going to talk about the increased expression of FHL2 promotes tumorigenesis in cervical cancer and is correlated with poor prognosis. The cancer is an abnormal growth of cells which tend to proliferate in an uncontrolled way and in some cases to metastasize. One of those is a cervical cancer that have some characteristics. First, it is formed in tissues of the cervix, that is the organ connecting the uterus and vagina. It is usually a slow-growing cancer, may not have symptoms, and one of the causes is the human papillomavirus infection. It is the fourth most commonly diagnosed cancer and the third leading causes of cancer death among females. Now I'm going to explain about 4.5 limb domain protein 2 that is the same like FHL2. The FHL2 is a member of the FHL protein family. It's an adapter molecule and can interact with many different proteins to play diverse roles in regulating cell survival, proliferation, differentiation, addition, and motility, as well as gene expression and signal traduction. The role of FHL2 in cervical cancer remains unclear. Numerous studies have reported that FHL2 may act as a tumor suppressor in a cell type dependent manner in human cancers. Previous evidence has demonstrated that FHL2 is overexpressed in breast cancer, gastrointestinal cancer, prostate cancer, and glioblastoma, though low expression levels have been observed in human rhabdomyosarcoma, liver cancer, and neuroblastoma. This study aimed to investigate the prognosis significance of FHL2 expression in cervical cancer and its effect on cell proliferation, apoptosis, and tumorigenicity via in vivo and in vitro analysis. Furthermore, we preliminarily explored the role of FHL2 and two possible molecular signaling pathway underlining its involvement in cervical cancer development. The materials and methods. The patients were 52 women of 39 to 71 years old with cervical cancer. The tissues were divided in two. One of the group of samples started at minus 80 degrees Celsius. The other group of sample was fixed with formalin and bathed in paraffin. The cell culture. The cervical cancer human cell lines were maintained in Dulbecos modified eagle, containing 10% fetal bovine serum in a 37 degrees Celsius, incubated with an humidified atmosphere of 5% CO2. The methods. The immunohistochemical used to identification of a tissues through a specific interaction between an antigen and antibody. In this case, was to identify tumor cells. The small interfering RNA and transient transfection is when introduce DNA or RNA into cells to generate change in the characteristic of the cells. 48 hours after transfection were measured protein expression, cell viability, and apoptosis. The other method is Western blot. Is when equal amount of total protein were separated by sodium dudecyl sulfates polyacrylamide gel electrophoresis. Then the protein are incubated with antibodies that react with the interest protein. In this case was to identify a specific protein from a complex mixture of protein extracted from cells. The last one is the apoptosis. It's a series of molecular processes that lead to cell death. In cancer cells, it may be locked. Later, the cell was analyzed using a FAC scan flow cytometer. Now I am going to talk about the results. In the first figure, the expression of FHL2 in the adjacent normal cervical tissues is less than cancer tissues. In the second figure, the FHL2 was upregulated in patient samples at protein level by Western blotting and used be acting like load control. In the third figure, the FHL2 protein levels was assessed in cervical cancer cell lines. The results showed that FHL2 protein levels were higher in Casqui and HeLa cells than in CIHA and C33A cells. In the fourth figure, to select the best FHL2 siRNA, the inhibition efficiency of three different siRNAs were determined in Casqui and HeLa cells. Compared with the control scrambled siRNA, all siRNAs showed significant inhibition efficiency in both cells lines. In the fifth figure, the analysis showed that Casqui and HeLa cells transfected with siRNA 955 had significantly higher levels of apoptosis than did NC388 cells. In the sixth figure, the FHL2 knockdown attenuated cervical cancer cells proliferation to further confirm the role of FHL2 in cell proliferation. FHL2 was transiently overexpressed in Casqui and HeLa cells and cell viability was evaluated. FHL2 was successful overexpressed in both cells lines. In the seventh figure, the FHL2 knockdown in cervical cancer cells caused reduced level of p-AKT and p-mTOR. The AKT and mTOR pathway plays an important role in regulation, apoptosis, and proliferation. In the eighth figure, the GFP detection via fluorescence microscopy showed high infection efficiency in HeLa cells by transfected FHL2 shRNA. In the ninth figure, the FHL2 protein expression was detected by Western blotting. The last one is the 10th figure. Is the Western blotting confirmed that FHL2 protein levels were decreased in xenograft tumor tissues. Discussion. Previous studies have reported that FHL2 levels are higher in numerous cancers than in normal tissues, and such overexpression has been associated with poor prognosis. Regarding the role of FHL2 in cervical cancer, Jean et al. Found it to be significantly overexpressed in the squamous epithelium of cervical cancer tissues, and the FHL2 regulated cervical cancer cell proliferation by facilitating MDM2, mediated the degradation of immediate early response 3. Numerous studies have shown that FHL2 is a critical factor affecting the biological behavior of cancer cells. In osteosarcoma, colon cancer, and ovarian granulosa cells tumors, FHL2 knockdown suppresses tumor cells growth, reduces cell viability, and inhibition cells migration. I have three conclusions. The first one is that FHL2 is overexpressed in the tissues of patients with cervical cancer. The second one is the FHL2 can have either a tumor promoting a tumor suppressing role in tumor genesis and development, and this dual role is depending the type of the tumor cells. And the last one is the FHL2 might be a potential target for the cancer therapy. However, more research is needed to confirm the specific role of FHL2 in different tumors.